Before we get into the nuts and bolts of B cells and T cells, I want to give a bit of an introduction to adaptive immunity. We'll talk about the different ways adaptive immunity can develop and the different ways in which adaptive immune cells see pathogens. There are a few characteristics of adaptive immunity that are distinct from innate immunity that I want to make sure to point out. The first is specificity. The adaptive immune system is much more specific than innate immunity. The adaptive immune system targets very specific antigens. That's why you can have, for example, antibodies that attack the type A surface antigen on red blood cells, but don't attack the type B surface antigen, because antibodies are part of the adaptive immune response, and antibodies are highly, highly specific. That's part of what makes testing for antibodies, or using antibodies to test for pathogens, that's part of what makes them so useful diagnostically. Second characteristic is versatility. The adaptive immune system is incredibly versatile. It can attack any of millions of potential antigens, and I might be underselling that. It could be billions, or even trillions. Not sure that anyone's ever calculated an exact number. But the B cells and T cells that you have in your body are extremely diverse, and they are theoretically at least capable, theoretically you have a B cell and or a T cell capable of responding to any particular pathogen you might ever encounter. This is different from the innate immune system. The innate immune system responds only to a set defined set of molecules. Third characteristic is that the adaptive immune system is systemic. That's systemic as opposed to not local. This is another contrast with the innate immune system. Innate immunity occurs at the site where the infection is, and it doesn't really spread. Adaptive immunity, you're protected no matter where that infection is. Say you get a staph infection on your foot, you generate antibodies against it, you generate a cell-mediated response against it. You're protected the next time you're exposed, even if that exposure happens on your hand. The fourth characteristic is one that we talked a little bit about when we started talking about immunity, and that's immunological memory. Remember that that's a characteristic of the adaptive immune response. The adaptive immune response to an antigen is both stronger and faster the second time you're exposed to that antigen. Fifth characteristic is tolerance. Tolerance is the ability to not respond to self-antigens. Remember, self-antigens are proteins that are found normally in our own body. They're not usually recognized by our own immune system, but they may trigger a response if they're transferred to someone else. Like if someone cut out their kidney and stuck it into your body, you are going to reject that kidney. Your immune system is going to attack it and destroy it, unless you are on heavy immunosuppressive drugs. So these 5 characteristics are all specific to the adaptive immune response that stand in contrast to innate immunity. Now, I want to talk a little bit about how adaptive immunity can be acquired. There are 2 ways. It can be acquired actively. In active immunity, your immune system is actually doing the work. You're exposed to an antigen, your immune system sees it, your immune system gets activated and learns to attack that antigen. There's also passive immunity. In passive immunity, your immune system actually isn't really doing anything. What you're doing is you're taking antibodies from the serum of someone else, another human, or maybe even a horse, some sort of large mammal that produces a lot of serum. You can take antibodies out of that organism against the particular pathogen you're concerned with and just inject them into your bloodstream. That gives you passive protection. A little bit of protection, that your immune system doesn't have to do any work for. Both of these forms of adaptive immunity can be acquired either naturally or artificially. In active immunity, natural active immunity is being exposed to infections. Right? You are running around on the playground, your best friend has chickenpox, you develop chickenpox a few days later. Right? Artificially induced active immunity is basically vaccination, where you take a weakened or killed form of a pathogen and you introduce it to the immune system so that the immune system learns to attack it. So instead of running around on the playground and getting chickenpox, you go to the doctor and you get your chickenpox vaccination. That's artificial active immunity. Passive immunity, there's both naturally acquired and artificially induced as well. Passive immunity is acquired naturally from between mother and child. So the mother's immune system produces antibodies that cross the placenta or leave in the breast milk in order to protect her infant. Artificial passive immunity you may not have encountered before unless you've lived in a place with venomous snakes or you've been bit by a strange animal. Because artificially induced passive immunity, that's what antivenom is, when you're given antivenom to protect you from a snake bite. That's also given after a rabies exposure, given after a bite. Rabies immune globulin is basically anti-rabies antibodies that can be given to help slow the virus down enough to protect you. Now, in active immunity, you typically get longer-lasting protection because you're activating all branches of the immune system. You're activating B and T cells. In passive immunity, you're not activating the cells at all. You're just transferring the antibodies, and antibodies only. And so since you're not actually activating any cells, your immune system isn't actually doing any work, you're only protected as long as those antibodies are around, but they're eventually going to break down and be lost. Now, there are two branches of adaptive immunity to discuss, and I'll have more detailed videos on each branch. Humoral immunity is what B cells do, and that's mediated by antibodies circulating in the fluids of the body. Cellular immunity, that's what T cells do, and it's called cellular immunity because it's actually the lymphocytes themselves doing the work. T cells and B cells are both lymphocytes, but they have some similarities and some differences. Both are formed in the red bone marrow. So remember, all blood cells are made in red bone marrow, including the lymphocytes. But T cells actually leave the bone marrow and go to the thymus. The thymus is where the T cells get an education, and that education is where they get a degree in tolerance. So what I mean by T cell education is they learn not to attack your own proteins, right? B cells get somewhat of an education, but they stay in the bone marrow until they're more mature. So they get some tolerance education in the bone marrow, but it's not as rigorous as what the T cells undergo in the thymus. T cells get a graduate degree. B cells, more like high school. Now let's talk more about this bit down here with antigen recognition. Both T cells and B cells recognize antigen. Antigen stands for antibody generating, and it's really just anything that the immune system can respond to. Okay, so an antigen is anything that can be recognized by a B cell or a T cell. So there are T cell antigens and there are B cell antigens. But B cells and T cells recognize their antigens in very different ways. T cells recognize it indirectly. The antigen is that little yellow squiggly fragment presented on the surface of another cell. B cells recognize their antigen directly. So this is a bacteria, this is the B cell, and this is the B cell receptor interacting directly with that bacterium. Sticking with the theme of antigens, I want to make the point that antigens can be quite large. When you're talking about an antigen, you might be talking about a single protein, but the term might also be used to refer to an entire bacterium. Okay? So if you're talking about a large antigen, an entire protein or an entire bacterium or an entire virus, these antigens, they have multiple sites that can be recognized by antibodies or by T lymphocytes. These diagrams are showing you antibodies recognizing multiple sites. T cells can recognize multiple sites on an antigen as well, but since the T cell antigen has to be kind of chopped up into fragments, you can basically imagine that there are multiple different fragments made from a single antigen that a T cell can interact with. Each individual site that an antibody can bind to or fragment that a T cell can recognize is referred to as an antigenic determinant or epitope. These terms are used interchangeably. They mean basically the same thing. It's the exact specific site where an antibody or T cell binds on an antigen. So for example, over here you have the triangle epitope, the round epitope, and the little square epitope. Over here on our bacterium you have the triangle epitope, the square epitope, and the round epitope. Right? Now each of these epitopes will stimulate different populations of antibodies and different populations of T cells. The name of the game here is redundancy. Redundancy is way better. The immune system loves redundancy because redundancy gives you better protection. Okay, it's basically maybe your body, your genetic makeup, maybe you can't make antibody A over here. But it's fine. You're fine. You're still protected against this antigen because your body does make antibody B and antibody C. Or maybe over here, your body can't make antibody B. It's just not in your DNA. You can't do it. But you're okay. You still got antibody A, and you can still attack that little square epitope as well. Redundancy. It's like having both airbags and seatbelts in your car. A backup system. Large antigens with a number of epitopes, and sometimes repeating epitopes, are called complete antigens. And complete antigens have two key characteristics. The first is they have reactivity, and that just means that they can interact with activated lymphocytes and antibodies. That means that they can be recognized by B cells and T cells and antibodies. Okay? But the second characteristic is immunogenicity. And immunogenicity is the ability to actually stimulate B cells and T cells, and to stimulate the production of antibodies. So reactivity is just the ability to interact with B cells and T cells. Immunogenicity is when an antigen can actually cause the B cells and T cells to respond, can actually trigger an immune response. All right? Haptens, these guys that you're looking at here on this slide, haptens have reactivity, but they don't have immunogenicity, right? So haptens are referred to as incomplete antigens. They can interact with a B cell or a T cell, but they're really just too small to look like a threat to the B cells and T cells. They're too small to stimulate an immune response. So usually these haptens are very small molecules. If you have a penicillin allergy, you're allergic to a hapten. If you get really nasty reactions to poison ivy, you're responding to a hapten. So the question then is, if these are incomplete antigens and they can't stimulate an immune response, how do we get allergic to them? Because allergies are immune responses. And the answer is that haptens, some of them are tricky. Some of them bind to what are called carrier molecules in our body. These are just large molecules, large proteins that are normally present in our body that the hapten can basically coat the surface of. So when you have multiple haptens all over the surface of a carrier molecule, it looks like a complete antigen, and your immune system will respond. Okay, now let's get back to comparing antigen recognition in both T cells and B cells. So this is talking about how B cells and T cells recognize complete antigens. We'll tackle B cells first because B cells are pretty easy to explain. The B cell receptor is basically a membrane-bound antibody. So the B cell antigen receptor is just an antibody that's stuck in the plasma membrane of the B cell. Right? So it recognizes antigen in the same way that antibodies recognize antigen, directly. No antigen processing required, no fragmentation required, no chopping up required, no other cells necessary. You could take B cells and mix them in with bacteria in a test tube, and the B cell would bind the bacteria. T cell antigen recognition is very different. You've got the T cell receptor there. It's that little blue protein. And on the other cell, you have MHC, and that MHC is holding this squiggly little yellow fragment. That's the antigen. So that's like a little tiny piece of one protein from that bacterium. That's what the T cell is recognizing. So this is called indirect recognition, because the T cell only recognizes that antigen after it's been taken in and broken down and processed into fragments, and then those fragments need to be offered up on the MHC molecule. And that's called antigen presentation. So the cell that's actually presenting, that's actually offering that antigen fragment up to the T cell, is called the antigen presenting cell. Let's take a closer look at that MHC. So here you're looking at, this is the cartoon version that I showed you on the last slide. Here you're looking at the actual molecular model of an MHC. So the MHC is this yellow ribbon model, and that little blue framework in there, that's your antigen fragment. So it just sits right in the groove of the MHC molecule. And there, this is a side view of that antigen fragment sitting on the surface of the MHC molecule. So it's like that antigen fragment is being offered up on a platter. If you think back to NK cells from the innate immune response, remember that MHC is the molecule that NK cells recognize the absence of MHC. And this is why some virus-infected cells or some cancer cells, why they downregulate MHC so that they can evade T cells. MHC stands for Major Histocompatibility Complex. It's also known as HLA, which stands for Human Leukocyte Antigen. But the name that I've been using, MHC, Major Histocompatibility Complex, that describes one characteristic of MHC, and that's that MHC acts as a marker of self. MHC proteins tell your immune system what's you and what's not you. That's why if you want to have a successful organ transplant, you need to carefully match MHC molecules or HLA molecules as closely as possible. You want them to be as identical as possible. I mean, you're never going to get complete and total identical MHC unless you're talking about identical twins, of course, because it's highly variable in the human population. But you want to get it as close as possible to maximize the success of the organ transplant. And this is a neat little figure that just shows what happens if you don't match MHC. So this is looking at transfer in mice of a skin graft. If you take a skin graft, a little patch of skin, from one mouse and transplant it onto a genetically identical mouse, so it's got the exact same MHC, the graft is fine. The recipient mouse can't tell that there is a graft there. The graft is completely fine. But if you take that same skin graft and put it on a genetically different mouse, and the same holds true for humans, that graft, it may be fine for a while, but it'll eventually die. It'll eventually get rejected. And the skin graft turns all necrotic and basically dies. So for a successful organ transplant, you match MHC as closely as possible, but you still will likely need to put the recipient on immunosuppressive drugs just to make sure that that organ survives. Okay, this is just an introduction, just beginning to talk about the adaptive immune system, and we skipped around a little bit, but you should be able to talk about natural and artificial active immunity, natural and artificial passive immunity, talk a little bit about the importance of the thymus in T-cell development, that education and tolerance, in self-tolerance role, talk about antigenic determinants, talk about complete antigens versus haptens, and compare and contrast how B cells see antigen and how T cells see antigen. Also talk a little bit about MHC proteins.